Hi everyone. So today's little video tutorial thing is on meiosis. So you would have done a little bit on meiosis last year. You may have forgotten, I'm not sure. So we're going to go over the basics today and as much depth as you need to know on the actual mechanism or process. Okay, so I've done my keynotes there for you. So meiosis, the main things, basics, it happens in the testes and ovary cells inside the nucleus. That's where the process of meiosis happens. It ends up, so your output from meiosis, the end product is 4 genetically unique gametes. And remember, a gamete is a sex cell, so it's either going to be a sperm cell or an egg cell. So they're not all new, they've just got, all 4 cells have got a different combination of the alleles in them. So they're genetically unique. Third point, each gamete has only 1 set of the chromosomes, not 2 sets. So a normal somatic cell has 46 chromosomes in humans, but the sperm and egg cell have only 23 chromosomes. Therefore, you have 1 allele for each gene. The reason that your sperm or egg has only 1 set or 1 allele from each gene is during fertilization, where the sperm and the egg fuse together, that creates the full amount of genetic material you need to function properly. Yeah, so you get the 2 sets when fertilization happens. So each gamete is, so the meiosis creates 4 unique gametes, and each gamete has only 1 set of chromosomes, so 1 allele for each gene. Meiosis reshuffles all of the alleles, okay? It doesn't create new alleles. The only thing that creates new alleles is mutations, which we've gone through already. All that meiosis does is it reshuffles what alleles are there. So it creates genetic variation by creating new combinations of alleles that we've seen before. That's why you look slightly different from your siblings and from your folks. It's just reshuffled the alleles, so they're sitting in different combinations. How does it work? So the mechanism is a flash name for process, and every time I think of mechanism, I think of steps. So I know that it's like a method or a step process that's going on. So step number 1, the chromosomes in our parent cell in the testes and ova replicate themselves. So if you have a look, I'll see if my mouse works here. Here's our homologous pair of chromosomes. This one was from our mother, and this one was from our father. They're homologous pairs. I know that because the centromeres are in the same place, and they're the same size, and they've both got the same gene in them at the same loci. Okay, step 1. Each of these chromosomes needs to replicate itself. So this chromosome here has gone bloop, and it's created a new one. And then this one here has gone bloop, and it's created a new one. The genetic information is exactly the same. Number 2. Crossing over then happens. So this is when the maternal chromosome, that's the pink one, I've done it very stereotype, where the pink one exchanges genetic information with the paternal chromosome, the blue one. Usually it only happens on these inside kind of chromosomes, yeah? The outside chromosomes aren't touching each other. And it will only happen within a homologous pair. So they're just swapping alleles basically, yeah? So that's crossing over, the exchange of genetic material between maternal and paternal chromosomes, homologous chromosomes. Step number 3, all of these chromosomes, so all of the pairs, remember we've got 23 pairs, yeah? They start to all line up in the middle or the equator of a cell now, but they line up in a completely random order. Okay, so this random order is called independent assortment, because the order that they line up in decides which gamete they go into. But we can't control that, it's random. So that's independent assortment. Number 4, once they're all lined up, the chromosome then gets pulled apart as the parent cell divides into 2. So that cell grows a new membrane in the middle, and now we're left with 2 cells. So it was 1 cell, membranes grown in the middle and between the homologous pairs, and it's now 2 cells. 1, 2. Okay. Step 5. The chromosomes in those 2 cells get pulled apart again. So you're going to get new membranes going. Yeah. So now we have 4 unique gametes. So 1 sperm cell, 2 sperm cell, 3 sperm cell, 4 sperm cell. The unique, can you see how they've got all different assortments of the alleles? So this one has all of the maternal alleles. This one has got most of the maternal alleles plus a little bit of the paternal allele. This one is mostly paternal with a little bit of the maternal. And this is all paternal. The separating of these alleles is called segregation. So let's recap. Meiosis happens in the nucleus of the testes and ovaries. It creates 4 genetically unique gametes. So they're all different from each other. They all have, each gamete has only 1 set of chromosomes. And the chromosomes have reshuffled. So they're all in different combinations than they were in the original person. Chromosomes, first of all, have to replicate themselves. And then the homologous pairs can exchange information between the maternal and paternal chromosome. Then they line up in a random order, so that's independent assortment. Then membranes grow in between them, they get pulled apart and membranes grow in between them, and the alleles are separated from each other, and that is segregation. So hopefully that helps. There's only really been 1 question, I think, in the last 8 years maybe, that's asked you to go right through the mechanism like that. But if you can remember it, it's actually quite an easy way of saying what crossing over and independent assortment are. But we will go into the genetic variation of how each one creates it with the next lesson.